Hi, everybody. Today we are going to talk about cellular respiration, and we're going to begin with a little introduction that talks about how we harvest chemical energy. So we are going to go ahead and get started. So one overall theme for today and the general point of cell respiration that you should always keep in the back of your head is that the point here is to make ATP. That is what we want to do. This shows that it's the mitochondria that's doing much of the work. The mitochondria is going to be a major but not the only organelle that we talk about. And so making ATP is the overall point. We want to make lots of it. And in order to do that, we need oxygen and we need food. So a little review on energy. Energy is stored in organic molecules like carbs, fats, and proteins. Typically, we're going to be talking about carbs and fats. We are specifically going to be spending most of our time pretending all we eat is glucose, although that's not entirely true. You can undergo cell respiration with carbs or fats and sometimes under certain conditions, proteins. Heterotrophs are organisms that eat other things. So herbivores, carnivores, omnivores are all heterotrophs because they get their organic molecules from food. And so heterotrophs have to have a digestive system in order to get their organic molecules. So those organic molecules are the raw materials to synthesize other materials. And those raw materials are going to be the fuel for energy to help us get ATP. It's a controlled release, so it does not happen all at once. So it keeps things in control, which is good for our cells. So we burn our fuel in a series of very controlled step-by-step reactions. Okay, so glucose is going to be our model, as I said before. We can, of course, harvest stored energy from other things besides glucose, but we are going to exclusively talk about glucose. This is a catabolic reaction because it's releasing energy. So we are catabolizing glucose to produce ATP. And this is the general reaction. So glucose plus oxygen is going to help us make ATP, and we're also going to make water and carbon dioxide as waste products of this reaction. And since this is an energy transfer, it's not entirely efficient. Some energy is going to be lost as heat. Okay, so just like in combustion is going to release a little bit of heat with the help of oxygen and some activation energy, respiration is going to be very similar. It's going to make energy. We're going to lose some energy as heat. We're going to need oxygen. And we're going to produce carbon dioxide and water as a waste product. Okay, so this slide is just showing you the difference between burning something, all of the energy being released at once, and what we're going to be doing with cell respiration, which is letting energy get released step by step. Okay, so how do we harvest energy from fuels? We need to digest large molecules and turn them into smaller molecules. So we have to break bonds, we have to move electrons from one molecule to the other. As electrons move, they're going to carry energy with them. Electrons have energy, and they hold that energy and move that energy with them. That energy is then going to get stored in another bond as the electrons find somewhere else to go. That energy can be released as heat, or we can harvest it to make ATP. So there are three places where that energy that's taken by those electrons from the bonds that can get stored in another bond. We can release it as heat, or we can make ATP with it. So in most of the examples we talk about today, one molecule is going to lose an electron, and that electron is going to move to another molecule. That means that the molecule that lost the electron becomes positive, and the molecule that gained the electron becomes negative. We say that the molecule that lost the electron is oxidized, and that gained the electron is reduced. You may remember from chemistry, you might have learned either oil rig, that oxidized is losing an electron and reduction is gaining an electron, or you might learn Leo, the lion goes grr, lose electrons oxidized, gain electrons reduced. Quick way of remembering that. Okay, so that is a redox reaction. Okay, so how do we move electrons in biology? Electrons can't move on their own, and in biology, they're going to move as part of a hydrogen atom. So that's basically a proton plus an electron together. There we have that, proton plus an electron. So when we move hydrogen atoms, not ions, but hydrogen atoms, we are going to be moving electrons. So as you can see here, we are, glucose is becoming carbon dioxide, and that is oxidation. We are losing an electron, and therefore we have lost hydrogen, right? Where did the hydrogen go? It and the electrons left. And they went to water. As we can see here at the bottom, oxygen did not have any hydrogens, and then it got reduced, and it picked up a bunch of electrons, and therefore it picked up hydrogen. So we see the carbon and the oxygen lost their hydrogen through oxidation, and the oxygen gained its hydrogen through reduction. But let's not think about it in terms of hydrogen. Let's only think about it in terms of electrons. The glucose lost its electrons, so it was oxidized, and the oxygen gained electrons, so the oxygen was reduced. Okay, so lecture check question. Electron carriers are important to cell respiration because they, A, control the transfer of energy between reactants and products, B, store energy through the production of ATP, C, allow for the maximum amount of energy release, or D, are the main product of respiration. Take a moment and answer this question. And the correct answer is A. They control the transfer of energy between reactants and products. They're not really very good at storing energy, so B cannot be correct. They allow for maximum amount of energy release. That's also not true, right? Because this is a stepwise reaction, so it should happen slowly, and they are not the main product of respiration. That would be ATP. So B, C, and D are all false. Okay. So redox reactions release energy as organic materials break down. Whenever we break a carbon-carbon bond, we're going to be releasing a little bit of energy. We strip off electrons from those carbon-hydrogen bonds. So when we turn glucose into carbon dioxide, we say that the fuel has been oxidized. And that is what happens to the glucose that you eat. It eventually becomes carbon dioxide. And what happens to the carbon dioxide after you eat it? It becomes, what happens to the carbon dioxide after it's formed? You exhale it. It's what we exhale. That's the waste product from cell respiration. The electrons are attracted to the more electronegative atoms, which in biology, that is going to be oxygen. I gave that away. Yep, oxygen is the most electronegative, which is why oxygen gets the hydrogen, because oxygen is more electronegative than anything else in this equation. So the hydrogens carrying those electrons go to the O2. All right, so we couple these redox reactions and we use the energy that gets released to make ATP. All right, so some comparison there. Oxidation, we're adding an oxygen and removing an H. But most importantly, you should think about it like that: losing electrons, releasing energy, exergonic. For reduction, most importantly, we are gaining electrons, we are storing energy, and it is endergonic. Okay, so what are these electron carriers that we've been talking about? NAD plus is one of them. NAD plus gains electrons to become NADH. And FAD is also one of them, which gains electrons to become FADH2. They look very, very similar. It looks similar to ATP, it looks similar to some nucleic acids, and as the penguin brought up, that's a mechanism of evolution, right? We built it once and we're able to use it in different ways. That makes it much more efficient. So we add the H there, and that H is carrying an electron, and so that is how electrons get added. Okay, so once we add electrons, you can think about it like putting money in the bank. We're storing energy in this molecule. So we have some energy saved up, and we're going to spend that energy later. Cell respiration is actually made up of 4 steps. So we have the anaerobic part. So anaerobic means without. The AN means without, and aerobic refers to oxygen. So that's the without oxygen part. So first of all, we have glycolysis. Glycolysis is respiration that doesn't use oxygen. And it happens in a cytosol, so it happens just out in the cytoplasm of the cell. Aerobic respiration, meaning oxygen-using respiration, happens in a specialized organelle called the mitochondria. So the mitochondria is going to use that oxygen. There's 3 parts that come after glycolysis: pyruvate oxidation, followed by the Krebs cycle, followed by the electron transport chain. So 4 overall steps. The first one is anaerobic, and the last 3 are aerobic. And we will find out how those work in a later lecture. So as we move on to learn about these different parts of cell respiration, you should always remember, what is the point? The point of all of this complicated stuff that we're going over is to make ATP. ATP is the point here. What is ATP? ATP is a modified nucleotide. It looks like that. The 3 phosphates are there. Okay, so this is the nucleotide adenine, and sometimes it's referred to as AMP. The M, you might be wondering what that stands for. That stands for monophosphate. If we add a phosphate, it becomes ADP, or diphosphate. And if we add another one, it becomes triphosphate. ATP has the most energy, AMP has very little energy. Remember, the energy is caused by the bonds connecting those phosphate groups, so that is where you'll find the most energy. Adding phosphates is endergonic, so those are very high energy bonds. They're holding those phosphates on. And if we release one, it's going to release a bunch of energy. Adding on those phosphates stores the energy because those phosphates are very unstable. Each negative PO4 is difficult to add, and there is a lot of stored energy in each bond. Most of it is stored in that 3rd bond connecting the 3rd phosphate. And the 3rd phosphate is the hardest to keep on there because it is so unstable. It wants to go away. The bonds of the negative phosphate groups is unstable, spring-loaded, and it can pop off easily to release energy. Which makes ATP a great molecule for doing just that, for releasing energy. Okay, so ATP just became ADP there, and releases about 7.3 kilocalories of energy. that can fuel other reactions. Phosphorylation, which is released phosphate now, can then be used on other molecules to make those other molecules do some work. It can destabilize them, and an enzyme that phosphorylates things is called a kinase. Okay, so an example of phosphorylation is the very first step of cell respiration, which is when we begin the breakdown of glucose. Happens. Remember how we said that adding a phosphate was very unstable? Well, we can add a phosphate to the molecule of glucose here, and that is going to destabilize. We actually added two. That's going to destabilize the glucose, and it's going to cause the glucose to break in half because it's unstable. That's actually the very first step of cell respiration. Okay, so ATP is not a good energy storage system. ATP gets used right away. So we have it for a little while, but it's the currency of energy, so we use it pretty quickly. ATP is very reactive. ATP transfers its phosphates very easily, and it's really only good for short-term energy storage. Carbohydrates and fats are more stable and they are better for storing energy. All right, now we see that cell respiration is going to make ATP, and when ATP is made, ATP then is unstable. It is going to release its energy as it removes the phosphate, and this whole cycle will continue again and again and again. A working muscle recycles over 10 million ATPs per second, just one muscle. So that's a lot of energy that ATP is releasing. Okay, so as we wind down here, how do we make ATP? Well, basically, I'm going to kind of spoil the ending of cell respiration. Basically, what happens is that we have this enzyme called the ATP synthase enzyme. That's what this big, weird looking purple thing is. The ATP synthase enzyme has a little opening that goes through it and allows hydrogen ions to go through. And when those hydrogen ions go through, they turn this little part like a rotor. It turns around. It's called the conformational change. And as it turns, it is going to bond a phosphate to ADP. And as that phosphate joins the ADP, it is going to make ATP as a result. So it is the movement of hydrogen through this ATP synthase that makes ATP. But how are we going to get hydrogen to flow through this ATP synthase? Well, we need to make sure we have lots of hydrogens up here first. And that is everything that cell respiration does, makes that gradient. So we are going to find out how that happens later on in the chapter. Okay, and that is where we're going to stop for today. We're going to continue this later, where you're going to find out about the steps of cell respiration.